एजुकेटेड बाय MIT यूट्यूब चैनल में खुशामदीद। Question number 1. Which statement best describes an enzyme? Option A, a lipid used for energy. Option B, a biological catalyst that lowers activation energy. Option C, a hormone that controls growth. Option D, a structure carbohydrate. Right option. Option B is correct. Question number 2. The region of an enzyme where the substrate binds is called the option A active site, option B allosteric tail, option C peptide bond, option D cofactor shell. Right option is option A active site. Question number 3. Which factor generally increases enzyme activity up to an optimum? Option A, removing substrate. Option B, extreme pH. Option C, increasing temperature within the safe range. Right option is option C, increasing temperature within the safe range. Question number 4. What happens to most human enzymes at very high temperature? Option A, they form DNA. Option B, they become carbohydrates. Option C, they always become faster. Option D, they permanently denature. Right option is option D. Option D is correct. Question number 5. A competitive inhibitor uses usually option A increases enzyme synthesis, option B bind to the active site, option C converts enzyme into lipid, option D destroys the substrate. Right option is option B. Option B is correct. Question number 6. Which molecule is commonly an inorganic enzyme cofactor? Option A glucose, option B DNA, option C starch, option D Mg2. Right option is option D. Option D is correct for this question. Question number 7. The protein part of an conjugated enzyme is called the option A apoenzyme, option B coenzyme, option C substrate, option D prosynthetic group. Right option is option A. Option A is correct, apoenzyme. Question number 8. A complete active enzyme containing of apoenzyme plus cofactor is a option A ribosome, option B zymogen, option C alloenzyme, option D polymerase only. Right option is option C. Option C is correct, alloenzyme. Question number 9. Which graph best presents enzyme activity as pH changes? Option A always 0. Option B straight line forever. Option C no change at any pH. Option D a single optimum peak. Right option is option D. Option D is correct, a single optimum peak. Question number 10. What is the effect of increasing substrate concentration when enzyme concentration is constant? Option A, rate rises and eventually reaches a maximum. Option B, rate always falls. Option C, enzyme becomes DNA. Option D, pH becomes 0. Oh, the correct answer is option A. Rate increases and eventually reaches a maximum. Question number 11. Vmax is best describes as the option A mass of substrate, option B pH of an enzyme, option C maximum reaction velocity at substrate saturation, option D minimum enzyme concentration. Right option is option C. Option C is correct for this question. Maximum reaction velocity at substrate saturation. Question number 12. Km is the substrate concentration at which reaction velocity is. Option A 2s Vmax, option B half of velocity max, option C 0, option D equal to enzyme mass. Right option is option B, half of velocity max. Question number 13. Which inhibition can often be reduced by increasing substrate concentration? Option A thermal denaturation, option B destruction of enzyme, option C irreversible denaturation, option D competitive inhibition. Right option is option D, competitive inhibition. Question number 14. Allosteric enzymes are regulated when a molecule binds. Option A at a site other than the active site. Option B only to DNA. Option C to the ribosome. Option D to a phospholipid bilayer only. Right option is option A at a site other than the active site. Question number 15. Which enzyme catalyzes the breakdown of hydrogen peroxide? Option A pepsin, option B catalase, option C amylase, option D lipase. Right option is option B catalase. Question number 16. Pepsin functions most actively in the option A alveoli, option B natural cytoplasm only, option C strongly acidic environment of the stomach, option D strongly alkaline blood. Right option is option C strong acidic environment of the stomach. Question number 17. Which statement about enzyme is correct? Option A they work only at 100 Celsius. Option B they change the equilibrium constant. Option C they are consumed in equal amounts during every reaction. Option D they are not permanently consumed by the reaction. So right option is option D. Option D is correct for this question. They are not permanently consumed by the reaction. Question number 18. A non-protein organic helper molecule for an enzyme is a option A nucleosome, option B coenzyme, option C polysaccharide, option D substrate only. Right option is option B coenzyme. Question number 19. Which condition is most likely to cause enzyme denaturation? Option A extreme pH, option B adequate substrate, option C optimum pH, option D mild temperature increase within optimum. Right option is option A extreme pH. Question number 20. Why are enzymes highly specific? Option A they contain only glucose, option B they are all identical, option C their active sites have specific shapes and chemical properties, option D they never bind substrate. क्वेश्चन नंबर 20 का जवाब कमेंट में बताइए। इन सवालों में से आपको कितने क्वेश्चंस के आंसर आते थे कमेंट में लाजमी बताना। वीडियो को लाइक और चैनल को सब्सक्राइब करें ऐसे मज़ीद मालूमाती वीडियोस के लिए।